Stalin was really into them and everyone used them. And then the West found antibiotics, used them, but nothing changed for the Soviet Union side. And you always have to dance around it with the historical side because it was like commie science. And so there was this bias that grew as well. And that's like one of the reasons that the fields have diverged.
And then John Iredale said, okay, let's take this seriously.
Let's do it. And it was completely because of his credibility. He wasn't just like some new doctor who's like, I think we should have phage. He was like, it's about time. And like, I need a legacy at this point. I don't know.
Your system's good for something.
Other doctors would find out just because they know John. They would be like, hey, I've got a patient. There's this guy named Tim from like 4 hours away in some rural area. And then once we took one Petri dish from him, we got like a new one every week. He's like, this is another guy. We're like, is it the same guy? And they're like, he's like, no, I don't think so.
We could treat all of the patients who like reach this last line of therapy and they're going to get better 70% of the time, 85% of the time. That seems worse, but we've cut this whole operational step and this point of failure out of our trial design.
Not having abandoned the phage field yet. I just am not convinced that it's not doable, and I feel like it's an execution issue, which I feel like is very exciting if it is. The more I talk to people, they're like, not giving me a satisfying answer that says like, of course, it's that.
Yeah.
Like no one says phage therapy's doomed. And I'm like, well, then I can't stop working on it.
Welcome to Development and Research, where we talk about people doing things differently in the world of clinical trials. I'm here with Jessica Sacher, who's a phage therapy biologist, for whom my first question is, what?
I've never even heard it described like that, but I like it. A phage therapy biologist. Yeah. Well, I started as a biologist who realized that the phages I was studying could be used in people, and then started going down the road of figuring out how to do that. And that's why I'm a phage therapy biologist. I'm going to own that.
So let's rewind 1.5 steps. Scary sounding word. What are phages?
Yeah. Phages are viruses. So they are scary sounding for that reason, but they are the ones that kill bacteria. So they're not the ones that kill human cells or animal cells. So bacteriophage is their full name, bacteria eater. And they look like little lunar landers. They have like a head and a tail with like spider legs. Yeah, and they're pretty huge compared to human viruses. They're just massive and they float around.
And this is abnormal on the microscopic scale. They're like everywhere around us. Like the bacteriophages are in the room with us now.
They're mostly in our bodies, but they're definitely everywhere because everywhere people seem to sample, you can find them.
The same way we find bacteria everywhere.
Exactly.
Like, there's this entire ecosystem of bacteria around, and that ecosystem includes bacteriophages.
Yep. Anywhere there's like any kind of moisture and any kind of food, which is like on the microscopic level, just everywhere. Maybe not so many on the floor. Like if I swabbed the floor, I wouldn't be surprised if I found nothing. But our feet, our shoes, I bet there's probably stuff in like plants, the soil. They're just like soil, water, and any kind of microbiome. Like they're just living amongst their bacterial hosts.
So when we're talking about phage therapy then, what is it trying to treat? How is it making humans better? And how is it different from just being exposed to the world around us?
So phages, are being used therapeutically. So it's kind of intuitive. And this is why it was discovered in 1915, basically. As soon as it was figured out that, there's like bacteria growing in a petri dish and there's like holes in them, it seems like. And that figured out that's something. Like we didn't have the microscopes to see it. We didn't have the genome sequencing to know that it was like a DNA containing virus, but we could see that it was killing bacteria. And so that was put in like rivers and people, like to save kids from diarrhea, because people just surmised that if there's bacteria and we think that might be causing disease and this is killing the bacteria, maybe we give it that and that will treat it. And it did sometimes. And so we're 100 years later, but it's still kind of the same process. It's just like the idea of purifying a little bit of the right kind of virus. And then anytime you want to kill a certain kind of bacteria, you just like combine them and you use it like a drug.
So this story starts very similarly to the story of small molecule therapeutics, where it's like, we find holes in our petri dishes and sometimes they're fungi that are like making weirdly shaped molecules. I guess we didn't like understand that at the time. First, it's just like, here's some stuff that's being made by fungi. If we grind it up, people take it, they get better. We've since learned, okay, yes, the thing that's happening is this molecule that binds the ligand, binds the protein and stops something from happening. so too we started from this, what is happening in nature when these bacteria are being killed through to today. But there the stories start to come apart because small molecule is the default, I think, mode of pharmaceutical development. Sometimes we have to remember that there are things besides small molecules, and sometimes we need to remember that there is such a thing as phage therapy. So where do these stories start to diverge?
There's so many people who've written about the history, and it's like also it's become like a trope. Like every phage researcher, the first paragraph of we just have to do the same paragraph. It's like the rite of passage.
Okay, so get another paragraph.
100 years ago, phages were discovered, and then they were abandoned when antibiotics were discovered because antibiotics work so much better. They're much more broad spectrum, so you don't have to be right about which one. It's much harder to be wrong. Antibiotics will just kill most bacteria, and especially during the phase of like the world wars, there's all these infections on the battlefield, and there's much more need to have some kind of way to combat them. And antibiotics were just so much more scalable, and they were also discovered around those same decades. And so, but phage therapy did keep going behind the Iron Curtain. This is back to the central paragraph of phage. And so, like Stalin was really into them, and there was like just a lot of use of phages. Everyone used them, and then the West, quote unquote West, found antibiotics, used them, but nothing changed for the like Soviet Union side. And so they kept using phages just like they had, and just like people had little vials of phages like on their belts, and they're like the soldiers did, like pour them on a wound and things like that. So there's a lot of, you always have to dance around it with the historical side, because it's like depends on who's writing it, but there's this idea that like it was like commie science basically, and so there was this bias that grew as well, and that's like one of the reasons that the fields have diverged and stayed diverged. As it just took a while for the West to kind of like run out of utility of antibiotics, and then be like, wait, there was something we had that was like very targeted. Now we need something targeted that works when these ones we have fail. And then, the rest of the world was like Russia and Georgia and places have been using them in clinic the whole time. So roughly that's the history.
Yeah, I think moving from a world where enormous sources of infection, or these battlefield infections, no one's testing anyone, no one's culturing anyone, no one's seeing the inside of the clinic, to today where in the developed world, we think of people fighting serious acute infections like, okay, well, now you're going to see a specialist. Specialist's going to like take some samples, culture some stuff. We can at least find out what's going to work for you as part of like an actual like clinical treatment, not just have to keep it on my belt when I get cut.
Personalized medicine, like it's a thing now.
Right. And personalized medicine is increasingly buzzily a thing. How does that then translate into sort of what the landscape looks like for phage therapy development today? Like, what is driving the interest in it? As you say, we're reaching the limits of antibiotics. How is that playing out, and what are the challenges that it's facing?
Yeah. So what's happening is that there's starting to be more antibiotic-resistant bacteria that are just in the hospitals. They're in the patients. So infectious disease specialists, when they look at bacteria, they have their certain list of antibiotics to use, and, the penicillins of this type and the cephalosporins, and there's, a list, and it's kind of like there's 20 classes, like roughly.
Mm-hmm.
It's not hundreds. And they will use those.
And for bacterial types, some of them just don't work against that type, and we go down the list, and sometimes we get a mutant where this one doesn't, but this one does.
And you go to the next one, there's like first line, second line, third line, and that, as I understand, changes like hospital to hospital, like region to region. It's almost like somebody decides like, okay, for now, this is the up-to-date one that Red Deer County Hospital is going by. It's like, use this one if you have this, and this one if you don't, if it doesn't work. So they go through their little roster, and then they're always getting more toxic as you go down generally too, because you want the safest, most effective ones first, presumably, and makes sense. But then there's stuff like colistin, which causes like kidney disease, and you really don't want to use that unless nothing else is working. So infectious disease physicians around the world at like the most major hospitals, like University of Pittsburgh transplant unit, that's when I came into the phage therapy space. They realized that like these physicians that have like apparently like all the resources you might want are actually out of options completely. And so their patients have nothing left. There's nothing on their roster that will work, or the only things left are either going to be way too toxic for the patient or the patient's allergic to them, whatever, no option. So that's the space where phage therapy is filling, is like all these patients that have like antibiotic-resistant bacteria. And some people say patients will get confused, like, oh, I have antibiotic resistance, like I'm resistant to antibiotics. And that's a big like misnomer, because it's not you, unless it's an allergy. It's the bacteria that's in you that's causing disease isn't susceptible to any of the antibiotics. And so that's where we have phages. And then the second part of your question about the personalized medicine landscape and what that looks like with phage is really about picking maybe something that, not because of this roster, but because of this patient and what they have. For example, a patient might have a lung infection, like pneumonia, and you culture it, you send it for cultures, the doctor orders the cultures, the person comes and gets a swab, and they grow it in the lab, and they find out, oh, it's Pseudomonas aeruginosa. Okay, common lung pathogen, yeah, that's the one. Then with phage therapy, you would need to take that Pseudomonas, that exact one, not just the word Pseudomonas. You have to
that one is genome, it's
have it growing on a petri dish. And then you have to have phage on hand, hopefully multiple phages, because they're so specific. They're like, this is their Achilles heel and it's their biggest strength. Like one phage will target like that Pseudomonas aeruginosa, but not that Pseudomonas aeruginosa. And we don't know what their rules are. And that's an active area of research, and we don't know. If you had 50 Pseudomonas phages or 1,000 or 5, you would test them each, pipette them down onto the petri dish, just like the 1915 like petri dish with the holes. We literally do that assay, and we this loosely, because hospitals do not have this capacity right now. But if that doctor is like lucky enough to know a phage biologist and like have a patient who's stable, who can wait long enough for him to mail the petri plate, literally like wrap it in wax, and put it in a plastic bag and mail it to me, a phage researcher at an academic institute,
so that you can like
so I can put my phages and see if any of them works. But it's amazing, because when they do work, we just like, there we go, we have the drug, and it will probably work if we put it in the person. And so maybe I'll just pause there, but yeah, that's what's happening.
So just set the scene here with scope and scale. Like, this doctor is lucky enough to know you. They take a culture from their very patient, wrap a petri dish with the culture, send it through the mail to you, and you're going to reach to your library of 12 or 1,200 phages to start this testing process.
So when I was doing this the last 2 years in Australia, I was like occupying this function as the phage tester for doctors, basically.
For the country of Australia.
We started, this is a whole other story, but a new experiment in doing this. We had 50 Pseudomonas phages. And we had 50 E. coli phages, and we had like 2 Staph aureus phages, and then we had a smattering of others. But like, it was not a huge library, but that's kind of like roughly how many we had, at least.
And those, you're just going to test all 50. Like your researcher is going to be like, yep,
just
got a sample, check them all. And then of those 50, for a good day, you'd get, or a good patient case, you get how many hits? Most of them were misses. Most of them were like, some of them were runaway successes. Paint us that picture.
I would say like we would get something like 5 hits out of 50. And we had no idea what to expect either. Because we found these phages from the fridge. They were not like in a nice bank, like ready for testing. Like they were someone's PhD project from like 10 years ago sometime. We just kind of went into this academic lab's fridge. And so we did not even know if some of these were redundant with each other. Like we didn't have time to really, and we didn't do this as the first step, check, like characterize them all. We were like, here's our Pandora's box, we called it, of like mystery Pseudomonas phages.
You just like have a numbering system or serial numbers.
They just had something on Sharpie on the top of the vial, some kind of like letter number code. And like, we hoped that it would be in some Excel sheet somewhere, but like, we'll find that later. We just checked.
When we need to write the paper or the case study or what's it.
And so, but we get like 5, or sometimes we get like 2, but we often did not get 0, which was shocking. I think we thought we would need 1,000 to hit this like patient isolate. But for some reason, I don't know. I still don't know, actually. Were we lucky? Is that normal? But that was our collection for Pseudomonas.
But like most of your patients, the vast majority of your Pseudomonas patients, like within this library of 50 that wasn't particularly designed, it was sort of compiled through the results of people's leftover research, the vast majority of those patients had phages that created little holes in the petri dish on their culture. Then you'd send 5 separate samples, or mixed on the other end, and then for a lung disease, the physician on the other end is giving it as an inhaled treatment or
Yeah. So the steps that happen between that part, because like that's where you'd want it to go for sure.
Yeah.
And the one thing that we didn't have yet was we just had like literally like 1 milliliter of each. And we don't, and it's probably not pure. It's like maybe it's like a mixture of phages. Like we hope it isn't, but we ended up finding sometimes it was. So we later had to, divide those up and, but also like they were not purified of bacterial debris yet. So the whole next step of the process, if we found our 5,
because they were grown in bacteria and it's like exploded out of the bacteria, but then we still have all the bacteria sitting around in our 1 milliliter solution.
Yes. And so we call it a lysate, phage lysate. It lysed, and it's the lysate.
Yep.
And so there it is. We, you sterile filter it. So it's like the holes are small enough that living bacteria are taken out,
Great.
But everything else is there. And so there's still very much chunks of bacterial cell wall. And that's very, very dangerous. And that's actually the most dangerous part of phage therapy is these elements of the cell wall called endotoxin. That's like part of the lipid layer that is on bacteria. And so that will cause like, at worst, something like anaphylaxis, because the human immune system really wants to find, like, it's trying to save you from bacteria.
Right.
That's one of its sensors, is that. So,
and when it finds like all these raw pieces of bacteria lying around, it's like, oh no, like a lot of work here.
Inflammation, and just like figure it out. And so you can certainly, like, what was keeping me up at night was getting rid of that so that we wouldn't kill somebody, because it was literally could kill them for sure.
So I, know only a tiny pieces of about phage therapy going in to do my research for this podcast, and my understanding and sort of like osmosed into me from the rest of the community is like, yes, that's the thing you can try, but like the major problem with it is sort of deliver a bacteriophage. It kills all the bacteria in someone at once, and then we have this like immune reaction to all the like immediately lysed bacterial problem of the bacterial that it's having the effect of. Is that slightly the wrong picture? And we should be more worried about the lysate that's coming in the injection than the like.
That's a really good point, and I think those both should be considered equally. But also they're sometimes lumped together or not considered at all. Mm-hmm. But the lysate, the reason, so it's present in the lysate, we got to clean that up. And you can do that with a series of purification steps and filters. And that's, I can get into that, but some of the work.
And some of the early trials failed to do that, which I think where we get our scariest negative results.
We need to talk about that. That is a thing that we need to clean up. But also the actual, like the release of the endotoxin from the target bacteria. Like this is what you want to happen is the target bacteria is exploding inside. When you give the phage, it goes and finds its target. Yeah. Injects its DNA and explodes it. Yeah.
We explode all the bacteria in here all at once. Sounds like a great picture. And maybe not all at once. It feel like your body needs some time to process the endotoxin bacterial parts that are being released.
And I feel like that is not as big of a problem as you might expect, because it's like a very valid thing to be worried about. Mm-hmm. And I am worried about it. Mm-hmm. But also we just don't see people having that many negative reactions to phages, even when they're given as unclean preps, even when they're given clean and they presumably work because the clinical outcome got better or the microbiology got better. Like, you see less Pseudomonas. So presumably they died. Right. But we're not really, like, phages have just been resoundingly safe for the most part, as far as safe goes. Like, I mean, if they have like some kind of reaction, it's like passes soon and they don't die for sure. Like there's no one that's died of a phage. I wonder about that. I'm like, what's happening? And like, why are we so worried about it here if it's not happening there? But the numbers could be way off. Right. And but they're both valid.
So you're doing the prep. You have this research sample, which is just this lysate, and like you got to get the endotoxins out because those you don't want to be immediately injecting in people. Yeah. I've read the method section of some of these papers, and like, oh boy, is this the like most involved method section of like how we prepped the samples, we like centrifuged this, we like went through a this reduction column, we went through this dilution, we went through this, and these are just like the steps that like you are doing at the Phage Center of Australia because one patient is got this.
Yeah. And so we had to come up with how we're going to do that, because it's one thing to be like, okay, if you're a company that you're making a drug, you're going to invest in your own like system and of producing the phage, and it's going to be like good manufacturing practices certified, like, and so you're going to whatever have your maybe a bioreactor, maybe you're making a lot of it because you're only making one thing, so you're going to invest in everything like that. But we were doing it at a case by case basis, and that was like going to be the case for a long time. We're not just like doing a one-off, but really developing a drug. We're like, how do we set this up so we can like keep doing one-offs? So, but also we were in an academic lab, so we didn't have any real budget to do anything or buy any machines. So kind of coming up with the like academic version, but still like the clean product at the end was like my task number one. But there's luckily, because it's such an old field, the literature is just full of like methods that you can try and things that are very cheap, like octanol, like an organic solvent that you just mix 1-to-1 with the phage lysate, and then it'll separate into its layers, and you just take the aqueous layer and most of the endotoxins out. I mean, you can dialyze that with like some dialysis tubing, and that's good enough actually. So we could do that.
So the methods are very established. They've been established over a century of research in former Soviet parts of the world, at least. But now we have this still decidedly non-GMP, process, which is like you've been there, pipetting in your sterile hood, and we're going to give this to patients because, in what medical context has this become an okay thing to get to?
Right. It's the realm of compassionate use, or various other versions of the name. Like we had a channel like that in Australia. And so it would be anyone who you only qualify for phage if you've tried standard of care and it failed. Mm-hmm. And your doctor.
The standard of care, the whole checklist.
Yeah. Like, and they have to make, it's case by case again. It's like they have to put their medical license on the line, the doctor. They have to like say, I have tried everything. And maybe standard of care is surgery, because sometimes like a prosthetic joint infection, and like the standard of care is to go scrape out all the biofilm and dump in antibiotics and then fill it back up a couple of times.
Great.
And this is happening. And so if whatever standard of care is, they've done it. Yeah. And we still have this chronic infection, recurrent infection, or life-saving infection. Yeah. And then that's the only way they can qualify for this sort of like established path for like drugs. The doctors can go reach for drugs that aren't approved if there is nothing else.
Yeah. But this is like very much a like physician's discretion, and like physicians sticking their neck out there for their patients.
Yeah, definitely. And their hospital, their employer, is actually the one who is liable.
They're taking their pocketbook out there in the
And, and so often the doctor might want to, but they have to convince the person. Yeah, the buck stops with the hospital committee, whoever decides. Like, can our doctor guy that we employ go and do this risky thing? Are we likely to get sued because we pay his insurance? Yeah.
Okay, so within this, like, our patients have so many ways. They've got a doctor who's familiar with this field, who hasn't written off this field because of its connections with communism, with Stalin, who's connected with this network of academic centers that do this service, phage testing and preparation service. They've gone through their whole standard of care. They've gone through their hospitals, risk management, experimental treatment committees. They've gone to you for the phage testing. That's the easy part, I guess. Mm-hmm. They've got these hits. You've done a bunch of work, but it's work that's procedurally established, and you sort of know how to do it. And I hope you didn't do all of them yourself. You're like training your researchers to do it.
There were two of us. Oh. We did one patient a month for the first year, and that was all we did. But yes, like the next rendition of that is, they brought in a few more people, and like it's becoming slightly more of a machine. It's very manual. Yeah.
It's not in the context of like randomized controlled trials, but like, what's the typical story for these patients who have like been through the whole checklist, including these extremely invasive things, and the doctors have got their weight, put their licenses on the line? What happens to the patients?
Yeah. So this is really cool because there's a pretty high rate that something good happens for the patients at this point. Like if you can get the phage, if you can get all that done, as you just said, like you're way more likely to fail on that than the actual phage. If we find that like once you do get an in vitro hit and that phage, like and that goes back in the patient and everything happens, the patient like usually does better clinically and or does better microbiologically, like sometimes eradication of like they get to culture negative. So there's no culture that can be cultured, which is awesome when they were like previously like dying of that infection.
And not responding to anything.
Yeah.
This is like,
Yeah.
usually works.
Yeah.
After everything else has failed.
Yeah. So like 70% is the number that keeps coming up for case studies. When people compile like 100 case studies across multiple centers, it's like 70% had clinical improvement. And then like 60% had like microbiological improvement or eradication, something like that. And these numbers, I mean, people like say, yeah, okay, that doesn't tell you it's 70% effective, but it is the number
These are anecdotes, these are cases, these are, yeah. Open label, and we worry the placebo effect and all of this. But
Exactly. And when I was always wondering, and everyone wonders this in the phage field, like how many negative cases are there? Because they wouldn't be reported, because we all know you're not going to write a publication unless you really feel like it's important. And some people have.
Publish a negative.
Yes. I mean, we should all strive, but like the reality is it doesn't go to the top of the priority list. Yeah. But I was always wondering, like, is it very drastically over-reported, the positives? But then when I was like actually part of 14 cases in a row, and like 12 of them were like great or nothing. But like there was 2 that were nothing, and like 12 that were like pretty good or great. And there was none that were bad. Mm-hmm. I was like, okay, I would have seen if we were hiding the negatives. And that was pretty cool. I think I didn't convince myself until the first patient that I like made the phage for, and then like started seeing that there was no trust involved anymore. Yeah. That's when I finally was like, I think phage therapy could work. Yeah, because I kind of always wondered. Just there's so many voices in the phage therapy space that are like, there's a bunch of cynical people who tried to make a company work and then it failed. And there's people who are still doing companies and they're not cynical about it, but like they point to all these failures, and you have to wonder. I wonder, like, if something it seems so promising but just doesn't pan out for 100 years, like maybe it's not that great. So I guess I needed some like hands-on feeling that it was working. I mean, it could have been, a fluke, whatever, but at least
14 patients is whatever. Like,
yeah, like I just felt like, okay, I think it wouldn't be talked about at all if we didn't have like quite a high record of good things happening.
It's facing such headwinds.
Yes. That's the one thing it keeps showing. And that would be like a kid, like we saved this kid from getting her leg amputated, and like they were definitely going to amputate, and we definitely got it so she didn't have an amputation. And
no with the counterfactual, but we didn't need the control like for this case.
Yeah. And there's this guy like who was like wheelchair shopping because his leg was going to get amputated in the Mayo Clinic, and that's like a really cool story too. But it's not just like a one-off story. So yeah, you can get rid of pretty bad infections with a phage as long as it works in the test tube. Mm-hmm. And there's a lot more headwinds to get yourself the opportunity to have your bacterial culture tested. That is kind of like the hardest.
Yeah. I want to eventually talk about, the gap from there you are, the two of you mixing one culture a month for the patients who have brought them in, and like what is the gulf between that and like this is a thing which my doctor, who may not be particularly heroic, is going to treat me with. I think there's a whole gap there. I think first I maybe want to rewind back and like what did it take to get to the point where you were there doing preps in the lab, convincing like a doctor to take a chance.
Yeah.
What is it like going from 0 to 1?
Yeah. The Australia one wasn't the first, I would say. It was the first time I was physically being the chemist in the lab. But when I started Phage Directory, like the last semester of my PhD, that was in 2017. And I was like convinced by my partner Jan to, help people get phage therapy on the internet, basically. Great. And he was like,
we're like Uber for phage therapy going on here.
Kind of. And he was just, he'd been in startups and stuff, and he's like a software guy, and he was like hanging out with people, joining their startups. The startups would fail or be boring or like helping people get food faster, whatever. And he was just like, ugh. And then he found out my field was in need of a life-saving thing that was like in academic labs, and like academic labs are never talking to doctors, and doctors are not aware they need to talk to the lab. And he was like, yes, that's an actual like worthwhile use of my website making, software making skills.
I guess it's not Uber for phage therapy, it's Craigslist for phage therapy.
Yes, we didn't even have that. People were using Twitter actually, and they weren't. It's not like many people were. Like one person used Twitter, and we happened to see it. And we were like, oh,
We have that.
yeah.
Oh, lucky that you were there.
Yeah, completely. Yes. And Jan was like, oh, well, why don't we just put everybody that you know there's a lab on a list? And then like, why don't we just like try to get doctors on another list? And then if they want to ask for a phage, we'll just like email everybody. And yeah, that was the plan. And so we did this. And so at that point, that was like a University of Pittsburgh doctor that was like looking for phages for this patient, Mallory Smith. And she had had a lung transplant, cystic fibrosis, like it was really bad for her. And then she had this Burkholderia infection, and then they wanted to kill it with phage because they couldn't kill it with antibiotics, and they couldn't find phage. And
So they post on Phage Directory, like, looking.
They would have if we had it by then, but we unfortunately like found out about it because of this case, started the directory like, and 2 days later she died. So that galvanized us, and also everybody who was like on the list already. They were like, what? And also like journalists from Stat News like wanted to talk to us because they were like, oh my gosh, now we have like something that can help.
Yeah.
Like, and there was like Mallory's face and like her whole story, and it was like, luckily there's Phage Directory at the bottom. And we were like, what? And Jan was like, I'm quitting my startup. Like, I was like, my God, like, I'm not telling my PhD supervisor, like, I'm supposed to be writing my thesis. But I was like, so that's how pretty much bad it was, and like ridiculous. But the Mallory had a doctor who was willing to look for one for a phage, and most doctors were not. That's also the hospital that let her get a lung transplant when most wouldn't, because they'll say no to that too. So in 2017, I'm just trying to paint the picture that like 2017 to like 2022, when I started making phages for Phage Australia,
we went from Twitter,
yeah,
maybe, to phagedirectory.com or
yeah, Phage.directory.
Phage.directory.
It's changed a lot. And like, it used to be that if you're like luckier than lucky if you are a patient that convinces their doctor to try this. And now, and even 2022, it was like, it had just like become more normal for an infectious disease doctor to know about phage therapy in the US and in even Australia and like Europe. Europe needs a lot of credit for bringing this forward. Like Belgium has been just like the main driver. But yeah, patients, it's completely hit or miss. And it was usually like, we would start getting emails all the time throughout those years, those 5 years of like, patient emails us, wants phages, and we're like, well, get your doctor on board. Yeah. And they're like, I can't. She says no. And I'm like, send them this PDF. And they're like, I will. Thank you so much. No one's answered me. They were so grateful. It still continues like this to this day. But some of them would come back to me 2 years later and be like, I got my doctor on board. And I'm like, why? And it's the same infection. It's like, because these are sometimes very chronic, or somebody has like, yeah, like they've had surgery, okay, they fixed it, but it keeps getting back, and it's just, or especially CF, like just like constantly reinfected. So whatever it was, they are the ones driving it, and like the doctors originally were like, no. Then the doctors were like, okay, but only if asked. And then but then like the next shift was like, so the Australia thing came about because one of these cases actually worked, I should say, and that was
they made the connection, and then the hospital, the academic center said, we should actually do this.
Yeah, it was the research lab. It was like John Iredell is the infectious disease doctor who is also like, he runs a research lab. And so he was like, I want to treat more patients with phages like this. Like he used our system completely because fluke, like one of his people who he works with met us at a conference, me and Jan.
And so he remembered Phage.directory, and like he found some other lab who like sent him some sample that someone purified it.
Yeah, exactly. It came from Israel, went to the US to get purified, then went to Australia. And like, it was wild. We didn't think anyone would help Australia because the shipment costs would be too high. But like 12 labs said they would. Anyway, we treated this girl dandy. She didn't have her leg cut off.
And then John Iredell said, okay, let's take this seriously.
Let's do it. Yeah. Yes. And it was completely because of his credibility as like a pretty senior infectious disease specialist. He wasn't just like some new doctor who's like, I think we should have phage. He was like, it's about time. And like, I need a legacy at this point. I don't know.
Your system's good for something.
Yeah. Yes. He had total like credibility, and like he did have like a platform to do this from. Yeah. And so he got a big grant for making phage therapy. It wasn't really called a center, but it was like 4 pillars that would be like banking phages, diagnostics, production, and monitoring, which would be like, no one's really been collecting the data very well, so we can't do real clinical trials. Yes. And this was like a big pioneering move on their part. It's like, we're going to do that. And they got money from the Australian government. And that was really exciting for everybody in the phage field, because they were like, nobody had been able to get the $1 million plus grants to do phage work anywhere. And so he got that. And then they were like, okay, Phage Directory helped us. How about Jess and Jan? Why don't you come like work with us somehow? Like, I don't know how. Just like, you could be a postdoc, you could be a bioinformatician. Jan's like, I'm a software developer. They're like, that sounds like bioinformatician. Yeah. And just come on in and we'll make this whole thing work. And the big thing they were trying to push was not just like a center, but like clinical trials, like a series of them. And the first one being this like STAMP trial, which is like a standardized treatment and monitoring trial for phage therapy. And so it's actually a case series, like they're on the patient level. Each patient still has to qualify through the compassionate use. They have to have tried standard of care, but they can have anything wrong with them because of that, because you're not excluding anybody. They finagled it beautifully, I think, because it's totally fits, but it's actually not how you would normally do a trial.
I guess it's this data collection system, which is existing independently from the existing system for getting patients individualized experimental, like last line salvage therapy care.
Yeah.
But we're going to collect the data.
Yeah.
All of us together.
Yeah. And the FDA has been to phage conferences for years. I've been listening to them give their talk, which is amazing. They come to our conference. Like, they send somebody and they say, please talk to us. We want to work with you. But they also say, don't collect data from compassionate use cases in series as if you're going to use that instead of a real clinical trial. Like, compassionate use is not a substitute for clinical trials as part of drug development. I remember this being hammered home. And I think what people interpreted from that was no data collection from compassionate use. Because, and maybe that doesn't explain it all, but that was what was thrown around, because everyone was like, wait, they're allowed to over there in Australia? And I think we were probably allowed to here too. It's just like, that's not a substitute, for if you're doing product development, you still have to do like a randomized trial.
This is how we're going to learn how.
Yeah.
This is how we're going to learn how to do the very first trial, is we're going to collect the data.
Which one should we do first? Yeah. Exactly. So the Australians, I should also credit Aline Katami. She's a pediatrician, and she's the one who was treating this little girl that we helped. And so she wrote this trial, and like she and John got it through, and they got the funding, they got the trial approved. They also got all the infectious disease doctors in Australia to sign something and say, this is the way that we're going to do phage therapy. All of us agree. And so that was so cool, because no other country, I've been running the Phage newsletter ever since I started Phage Directory, started that. And so I've been putting the coolest news out, 5 coolest news things every single week for 5, 6 years. And it's like, no, nobody's doing that. That was so cool. So they're like, yeah, come on over. Then COVID hit right when the funding was secured. Then the funding was frozen. And then we couldn't get visas, but we luckily didn't
exit Australia for a while there.
We weren't there yet.
Okay.
It was right before.
Oh, we'll set it up, and then
yes,
right.
We amused ourselves until then. We did other stuff. We just kept running Phage Directory. But and we were working toward everything. But by the time we got to Australia, we're ready to be okay, we have patients, and this is your actual question was what's it like for the patient, and I wanted to illustrate the difference. And the difference is, instead of patients trying to convince their doctor, well, John Iredell is the doctor who's running the trial. He personally has 21 patients at the hospital next door that need this. And we had all their samples because he's been collecting them. And so we just started picking.
You just land on day one. Just great. Yeah. 20 patient backlog. Get to work.
Jess, what do you think we can give this a go? And I was like, what do you mean? Because I actually didn't think I was going back into the lab. I didn't think I was going back into the lab at this point. I thought I'm going to be my Phage Directory. I'm going to be in the role of helping us get collaborators and helping us integrate with other phage banks and get more sources of phages. But then I was like, oh, my phage making skills are
actually pick out the tire and take the apparatus later and lower in the.
Yes. Okay.
So there you landed finally got your COVID visas, immigration, whatever. And Don said, okay, got 21 patients. Let's get started.
Let's go. Let's give it a go. And I was like, what does that mean? And he's like, well, do you think we could just purify the phages ourselves? And I think it wasn't at that point decided, because one thing about the STAMP trial, it's designed as, this way to get the patients, all the miscellaneous infections, you could treat them all, whatever. But they all get their own phages, and that was really cool. But
once the procedures, different sets of inputs.
Yes, exactly. Like same dose by the numbers, like 10 to the 9 phages per dose. And, same amount of doses in the same
purification column and the same this.
Well, the thing is, none of that was specified. Everything to do with making the phages, where they come from, how we make them, was not in there. It was just like, they will get phages if there are phages that, kill their bacteria. And they'll get this many of them at this interval. And we will check it by these methods. But like, where are the phages coming from? And I didn't really ask this question, I don't think, because I remember being surprised and then being like, I shouldn't have been surprised. So the phages are coming from me. And I was like, well, I roughly know the literature. I know who my favorite groups in the field are that I would copy if I was the one making the decision of what methods. And he's like,
oh, there's more.
I didn't know. And so he's like, give it a go. And so I gave it a go. And luckily the first patient that he had, he chose the first patient we're going to do was this guy who had this abscess in his chest where his lung used to be, because it was taken out from cancer treatment like years prior. And it was full of E. coli. It was like an abscess. And they couldn't get rid of it because I guess, the antibiotics, maybe they weren't getting there, but also once they're there, it was resistant. So, okay. So the goal was we're going to do intravenous phage therapy. So it has to be as clean as possible.
Yeah.
And
nothing, filtering from the body on the way in. If we have endotoxins, we're injecting them straight.
Well, we were going to clean out the endotoxins as much as possible. Yes.
You're writing the methods.
Yeah.
How are you cleaning out those endotoxins? Great.
I was like, okay, we'll use Optinol because I like this method. It's called Phage on Tap, and it's this researcher I like, Jeremy Barr. I think he's probably good. I've known him for a few years at conferences. I'm going to copy his method instead of the other methods.
Yeah.
And luckily, though, it worked out beautifully the first time. And this tends to happen in biology. It's like it works, and then it is way harder every other time. But beginner's luck can save you, probably because all the ones it doesn't work.
We just never hear of. Yeah, right.
Yeah. And so it often happens that you get this beginner's luck. And so that happened. And also we were lucky that it was E. coli, because that's the easiest organism just across the board.
To culture, to for phage model, it's
it's the fastest growing. Yeah. It's the model organism for bacteria. So it happens to be E. coli that's causing his problem. And we happen to have phages again that came up. We had our 50 E. coli phages. We found some, they worked. And then the Optinol worked really well to get rid of the endotoxin. We found a way to test the endotoxin levels very accurately. And that was crucial, which I haven't realized so much until now that I don't have it anymore. That we had the Sydney Cell and Gene Therapy Center downstairs in our building, which was again a fluke, but the Westmead Institute for Medical Research is where we were. And they happened to have people doing CAR T therapy in the basement and then phage therapy on the fourth floor. And we went, I don't remember, but I think I just talked to people until someone pointed me to, you guys test endotoxin. And the FDA and the FDA and the US specifies endotoxin limits for anything that you inject. It doesn't matter if it's phage or anything. And so in Australia's TGA, their FDA, they look to the same levels and they're like, hey, FDA says five endotoxin units. That's how many you can have. And so I found that there's an endotoxin reader downstairs in that facility and we can do it. So as long as we had the way to prove that the dangerous thing was very low in number, then we could we argued at least purify whatever method, as long as the dangerous thing is gone. And that's what we did. And then we treated the guy, and we also had luck with that. We cured his abscess completely. He became completely off antibiotics. He'd been on intravenous antibiotics for 3 years, the entire pandemic. He was on an IV, had to have that at home. And then he doesn't need it anymore. And I was like, oh, this stuff works. So it was wild, but that So those 21 patients, we started just testing through for all of those. But we also, the next thing, again, getting to your question of like the difference now in doctors' views as well. Like we started having like other doctors would find out just because they know John, like we weren't advertising it really. And they would be like, hey, I've got a patient. There's this guy named Tim from like 4 hours away in some rural area, but he's like, I got one. Like, and then once we took one petri dish from him, we got like a new one every week. He's like, this is another guy. We're like, is it the same guy? And they're like, he's like, no, I don't think so. And it was like, well, it's a Pseudomonas. Yeah, it's a different one. Okay. So like, it was like more doctor by doctor. But once we found a doctor that was like, once they're into it, they had like many patients. Mm-hmm. So I extrapolated from that like infectious disease physicians have many each right now that like patients that would benefit from phage therapy. They just do not have phage access, and it is very laborious for them to try. So they don't have time either. But certainly
they're not going to be pipetting.
No.
Obviously.
Yeah. So that's how it is. It's now no longer patient versus doctor. It's like doctor versus time.
Great.
So that's impressive, but still hard. Yeah. But it's so fields shifted. Like the physicians are on board now. Yeah.
Okay, so that brings us up to what feels like a much better world. There are patients who are coming off IVs. There are patients who are not having amputations. There are patients who have this thing and it doesn't work at all. But there are these cases, and not rare cases, where it succeeds where literally the entire medical toolbox has failed. There's still an enormous gulf between here and this is a like thing that doctors prescribe within like the standards of their practice, that things that gets covered by insurance, things that gets covered by Medicare in the US, by public health systems the world around. And conventionally, that gulf is crossed by commercial development. That there's a for-profit company, perhaps a for-profit company that originated at an academic center. It spun out of a university. It spun out of other sorts of nonprofit labs. We do have those examples. The precedent from something coming from the nonprofit world and saying, you need to be in the for-profit world in order to be developed. We'll transfer out the patent and some royalty rights come back to the institute, whatever. But that almost universally is the next thing that happens. Mm-hmm. You were talking through a development from 2017 to 2022, where like the sort of treatment landscape in Australia had gotten much better, and like the next mountain to climb is like, well, is this going to come to commercial development? Mm-hmm. And what have you seen in terms of like the movement on that? Like, is this going to come to commercial development, or are we just going to live in this like, yeah, there's a case series if you know John, or if your doctor like has had a previous like heard the talk of the where are we going?
I think this is definitely where we are. This is the question to ask. This is where we're at, is the struggle of like getting to that. And there's been, since the 90s, a lot of phage companies have started and failed. And there's like, everybody talks about like, there's skeletons on the road or whatever, go find out why they failed. But the ideal would be, or the like way that things seem to be going, is that, you get a cocktail of phages and you put them together, and now it's a broad spectrum phage cocktail. It's a drug. It's just like penicillin, which is a broad spectrum antimicrobial.
But you're like, no, I reach for the penicillin. Yeah. I reach for the phage cocktail. That's what I treat my patients. It's always the same thing every time. It's not this prepared one-off. And in fact, this cocktail was prepared, good manufacturing process, stainless steel bioreactors and all the rest. And so we're going to take your 50 Pseudomonas phages, develop them as a cocktail, and then we're going to run randomized phase 1, phase 2, phase 3, phase 3 again, trials to treat patients with this 50 phage cocktail as a commercial product against their bacterial last line bacterial condition. Like, that's the path to this being in the book instead of outside of the book.
Pretty much, although there's a few things that like make that really hard.
Yeah. So I want to dig into the it's not as simple as that. Well, what makes it not as simple as that, and like what to the point of the podcast like needs to happen differently.
Yeah. So one of the main things that makes that just jumps out is we can't have a 50 phage cocktail.
Okay.
And the reason
Why not?
seems to be, we should challenge all these assumptions. They made one with 12 and it died off quickly.
All
So
right.
that's the short answer. People think,
Like on the shelf, it degraded.
yes, while it was waiting to be used, as 12 seemed to go down in activity. So this is, I mean, that's the super short answer. That's one example. But the complexity of manufacturing. So the reason, like, we can get into like the shelf stability. Yes, that's a problem. And
Well, we have different components, and you can only combine them at the end. Is that, it's not our only problem.
Exactly.
Okay, so but like,
I would say probably the big reason why you don't have 50 is because manufacturing complexity would be so high, and to have like 50 different
50 different bioreactor production lines, and
yeah, it seems like that's a pretty big barrier. And even most cocktails are being developed with 3, 4, 5, 6, and that's like the highest that I'm seeing lately.
what's
Like-
the lowest number where it might work? Because every additional one is too much of a burden to
Yeah.
this unproven.
Yeah. And also like every phage, like I guess there's this tendency toward the minimal number that's like going to be the broadest. And you actually don't need 50 to get to like 80% coverage. You need like 3 often.
Sure.
It totally.
If you're doing it blind and just
Yes.
like, one of these 3.
Yeah. So I think if we needed 50, it maybe is possible that we actually could.
But if 3 is 80%, 85%
Yeah.
coverage, you pick your favorite
Yeah.
6. If you can get to your favorite 6, okay, fine.
Yeah. So that's roughly one thing. And the other thing is like, I mean, a lot of people put this forth, which is you still have the long tail, which is actually what we're dealing with now. Like we have most things treated by antibiotics and then the rest, okay, phages are zeroing in on that. But then like we're kind of doing the same problem as antibiotics. If we make a cocktail and we have that fixed, now we're going to treat like the 80% of that. And that's still the.
And it needs a compassionate use to
Yeah.
pull one off for the, don't do any of the other 47.
Yes. And so there's also a sense that like for all the work of doing a phage product, if you're going to do it as a fixed cocktail, you now do not have your advantage anymore over antibiotics, which are causing like resistance. And so you're going to have resistance and then what? You have nothing. And so people don't want to seem to put that forth. Like it's not, that's the pushback against the cocktail version. It's just like, okay, it's at best just as good as this thing that's not good enough. And fair. I think it's probably still going to be like, we still can hit a lot of infections with like that approach. And there probably should be, a Pseudomonas cocktail, a Staph cocktail, an E. coli cocktail, or maybe combined pathogens for like specific disease. It just depends. It's probably going to need to be like a bunch of cocktails on the market ultimately for across the diseases.
And across like the scale of decades, we're going to need the new Staph cocktail, the new Pseudomonas cocktail to come through the commercial development system somehow and show up before the other one is, been useless for decades and years as we develop this in technology. I mean, we see this problem in antibiotic development where it's like, we'd like to have the new antibiotics before we absolutely need them, but incentivizing that through a commercial process where everything is being chained back from like decisions by insurance payers at the very end and where the investors think that that's going to go. That gets real murky when you're trying to break new ground.
Yeah. So those are some of the issues, but ultimately it doesn't have to be crazily more complicated than that. Like you could probably, like that would be great if we actually got to that point where we just had like a few cocktails that were like pushed through and you check in advance that they're reasonably broad coverage for that disease type, or like at least type that's something that you can do to check that it will be the right cocktail. So like, I guess this brings in another aspect, which is
we culture it gram negative, or we culture it Pseudomonas, or we like there's some level that you're going to need to narrow down.
Yes. And I mean, there are like the country of Georgia, the Republic of Georgia, they have been doing phage therapy for 90 years.
So what's it look like there?
Yeah, they actually use cocktails that are like sourdough starters. And every year they like add to them, and then they keep like sampling the environment or probably whatever clinical isolates come in, and they're adding.
Just like, here's some more Pseudomonas, what grows on it?
Yeah.
Okay, we'll isolate that then.
Yeah. And then they're just adding to the cocktail. And so it's become this like, who knows how many phages, there could be 100, and yet it's like the cocktail is not for Pseudomonas. The cocktail is for lung infection, for example. I don't know if they actually have a lung one, but they have like a gut one called like intesti-phage.
Right.
And it's, you take it when your gut's having problems. And it's got like a few different pathogens are targeted by the phages in there.
Maybe in the West, I take Pepto-Bismol here. I'm just like, well, if it's an infection and not a toxin,
But they're just like, my stomach hurts, and that's normal. And so we could
before going to a doctor. This is all OTC. This isn't just the pharmacy.
Actually, so there's over-the-counter phages in Russia for sure. And there's, I think, over-the-counter phages in Georgia, but Georgia also has like a clinic,
prescription strength
hospital,
their prescription version.
I don't know, if the strength does change or what. I haven't been yet, but it's happening over there in a different way. I don't think we're thinking of it disease specific as much as pathogen specific, plus disease specific here. Yeah, but it could be simple. It's still like not like impossible to wrap your head around doing cocktail, and like there are some companies making progress on that.
I think this is fascinating because one of the things that we see when people are pushing the frontiers of like modalities of medicine is we say like, oh, here's a new, 21st century like sort of space age therapy. It's this super sophisticated. We need the most sophisticated medical centers to deliver it. We see this with gene therapy, as people are like, where do I go for gene therapy? Like, oh boy, I hope you're in the city of Boston or like maybe New York. Like the most specialized doctors in the world who have studied who have pushed the cutting edge of the research, those who are going to do your like $1 million gene therapy stuff. But this is not there. Like these clinical trials don't have to happen at the most specialized center in Boston if this treatment and follow-up and care of these patients is happening in the Republic of Georgia. It's certainly happening in Australia, but it happens in like a bunch of places whose like medical system is on par with that. If they can deliver 1980s era care, they can be delivering these. There's like not an additional barrier to the complexity of this seemingly new therapy. Yeah,
that's a really good point. And it's cool because like, people put it on a spectrum of like totally personalized to the max and like pill.
right.
And it's like, there's like personalized things that actually are like much less infrastructure requirements than you might think. I would say the one thing that does need to be there is some kind of currently phage lab, or there has to be phage microbiology going on. And currently
to do the testing, the sensitivity testing.
To do the sensitivity testing. Although
not for a cocktail. If you had a cocktail and you were running it through trials, you'd produce it somewhere in some phage lab and then you send it to some hospitals.
You're right, in that version of the world. Although I think it still remains to be seen if like cocktails will need a diagnostic as well.
Mm-hmm.
And I think, I don't know which way that will go. It seems.
No, I guess you culture sensitive to the cocktail at least.
Yeah.
Got it.
That's so.
Well, I mean, that gets to a question of trial design, I think, where if you're telling me that 85% of the patients reach the last line salvage therapy are going to culture positive to like one of the strains in this single one size fits cocktail, well, we could make the choice. Like this is a design choice of the trial that we're going to run, that we can say we're going to collect samples from the patient, we're going to culture them, we're going to need a phage lab, we're going to treat it with a cocktail, we're going to see whether spots appear in the petri dish, like they did in 1915. And only for the 85% of patients who do we go on to the next step. And for those, it'll be 70% effective. Or it would be possible, it would be operationally possible to set up a trial where we admit patients on, you've reached the last line of therapy, you actually have no other options. We're not going to wait this week-long back and forth, lost to follow-up amount of time to go through this culture and restrict ourselves to doing our trials only in places that have an attached phage lab. As their researcher who's going to be preparing these things on the bench, we could treat all of the patients who reach this last line of therapy, and they're going to get better 70% of the time, 85% of the time. Right? That we're going to have some 60% of these patients are going to get better, not 70%. That seems worse, but we've cut this whole operation staff and this point of failure out of our trial design.
And maybe that's why people, it's hit or miss whether you see trial designs, including a susceptibility testing step. Because I could see the argument for being like, nope, we don't want to know.
Yeah.
And then retrospectively maybe checking.
Right. If you decide you need to know, you can go this entire journey and upgrade your 60% efficacy to 70% efficacy in a subset of patients, and the remaining 15% of patients weren't treated, weren't given that false hope, weren't given that whatever risks of interaction with the system. I mean, there is a understanding of medical ethics that if we're treating people with things that are futile, we shouldn't do that. That's sort of outside of the practice of care. Where that comes up against well, how much work do you need to do to make sure it's not futile while you're not treating them? There's what did Hippocrates say about that? Right. about how much confirmatory testing you need to do before you treat your patients with the Phage cocktail. So I can see it both ways, and I can see it from the perspective of the trial designer, where you're okay, if I design a study and it's 60% effective, is that going to be compelling? Versus if I design a trial at 70% effective, it's not going to be compelling. On the other hand, maybe that restricts what countries you can do the trial in. Just whole geographies become not places where you can do trials, and that's the sort of trade-off between something which appears in the lab and something which appears in academic literature, and something which is only at the very last mile of what buildings are next to your hospital.
Yeah.
It is crossing these worlds in a way that I think both sides need to understand the other side, or whoever it is who's going to bring this forward, I think, is either going to make the right decisions, either going to make the right decisions. It's going to make a set of decisions here, and as a consequence, they're going to face a set of challenges, and their challenges might be that we have 60% instead of 70%. Of their challenges might be we have to do the sensitivity filtering, and we have to make sure we don't lose our patient to follow up every time. And also only get the patients who consent to the sample removed specifically for this, or to wait for this instead of the other thing. This now gets into the thing that I find fascinating is how does this work get designed? What choices get made? And what choices are going to need to be made here differently in this place than ways that we normally do them.
Yeah. No, keep going. I'm just rethinking my own field in very new ways. And it's new for me to get into the clinical trial design side. And I've just also been curious about how do clinical trials get designed? Because there's only 7 trials, 13 if you're generous, of phages.
Most of them are safety trials. We got our 3 efficacy trials, 2 of which worked.
Yeah, and so
one of which had a stability problem.
Yes, they all fail for a sad reason if you're trying to just get an answer.
Oh, there's a lovely chart from the paper which we'll put in the show notes, which is things that can go wrong in your trial are the columns, and it's X, X, X.
Yeah.
Two of the trials have all of them are green and succeeded. Green. It's just those 2. It's just the 2 that are green on all the X, and a little bit written for clarity for a fact, but just a little bit you just gotta get 8 things right in your trial, or else it's not gonna work.
Yeah, they describe in that paper a constellation of good things have to happen, and I'm is that how it is for everyone?
Probably just given to us by astronomy. We don't put it there.
Yeah. So I guess my central question in not having abandoned the phage field yet is I just am not convinced that it's not doable, and I feel it's an execution issue, which I feel is very exciting if it is.
Sure.
And
everyone keeps failing for reasons that we know.
I know.
Great.
And they seem they're fixable. I know I don't mean to disparage them or think I wouldn't have made that mistake, but
the first people out got the wrong thing, or the first people out they did their recruitment. Turns out that there aren't that many patients. You need to be recruiting more broadly than that. You couldn't be asking the patients to stay for sensitivity testing, or
or the Phage they thought that they thought the hospitals were collecting data on whether there was more than one organism in the burn wound, but they weren't. They were just collecting
at least this one, or this one, or
one. It's Pseudomonas, and they were like, "Great, our inclusion criteria must be only Pseudomonas." And then it's I don't know, we didn't mean that.
We meant that it's Pseudomonas. We wrote that down.
Yeah,
plus.
Probably other stuff in there for sure there is, and it's crap. We can't use those people.
Until they
So they didn't get
And
enough
so they didn't get
people.
enough
And it
patients.
took them so long that their drug went off waiting. And so just like, oh, and you know, the one in Bangladesh with the oral phage therapy that they did before that, like that was sponsored by Nestle. Like they were like doing that, and it was the phages were made against the pathogen they thought was causing the diarrhea, and then they were pretty sure it might not have been that one causing it in the end. And it's like, cool. And there's more to it, but like,
I mean, there's the other side of like treating like earlier on the diagnostics.
yeah.
Section.
So I just want to find out for myself, I guess, like what is the reason that these things haven't made it to, why don't we have a phage cocktail in the market?
Yeah.
Because we should be able to get that done. I probably just don't know enough about the realities of clinical development. But the more I talk to people, they're like not giving me a satisfying answer that says like, of course, it's that.
Yeah.
Like no one says phage therapy's doomed. And I'm like, well, then I can't stop working on it.
So I guess with that, what's next for you?
Oh man, I don't know. So
You're no longer in Australia.
Yes, I left Australia. It was really fun and I really miss it. And I just didn't want to live far from my family forever. So I had to like quit while I was ahead and be like, this is the next transition phase. You guys are hiring a bunch of new people. Like
The center's still going strong.
yes. Yeah, they have like lots more people.
More than 2 people per parent.
15 or yeah, more than 2, greater than 2. I'm amazed. Yep, they're continuing to treat patients. I think they're on like 40 now. Like we did like 12, 14, and then I left, and then so they're great, just cruising along.
Great. Appending to the case series, we're learning about data, we're learning about this.
Yes, I know that they'll probably start at some point trying to spin something out. Once they have a cocktail of phages that looks right, they'll try to do a trial. At least that was the plan. Like not this
a trial on the cocktail level.
Yes. Yeah. Like, okay, CF. What are the Pseudomonas phages? And they'll do an array of trials and then they'll try to commercialize those, I think. And like, yeah. And there's companies in the US doing the same. And in Europe, things are going. But for me, so I was like, well, at the time of wanting to leave and come back to North America, I was thinking, how can I get the same kind of job in my own continent? And I was looking around and the choice was like, okay, I could try to join one of these phage companies that's already going. But I thought if I did that, I would be like at the bench doing the pipetting, being the phage maker. And they already kind of have that taken care of,
Yeah.
That wasn't as exciting to me. I was thinking, okay, if the company is going to make the cocktails, who's going to do the rest of the long tail of all the other personalized cases that need to constantly be like treated too? Like, I think I see a world where it's cocktails and personalized phage therapy, like forever.
Yeah.
And there's a lot of budding interest in phage therapy centers around everywhere. And we started seeing these things pop up. And so people were always asking.
In the US, where previously it was just a phenomenon.
And in like Asia and like South America and even in Canada. And so that's where I'm from. So I'm like, otherwise no one would point it out. But there is interest in building phage therapy centers so to do what I just did in Australia, get that set up in other places. So that was what I was excited to do. And I still like, it's not like past tense. But I'm not sure if it's the right thing yet either.
Yeah.
But I joined a lab at Stanford, Paul Boyke's lab at Stanford Medicine. And I basically emailed him and was like, do you want to do anything with phage therapy space? And he was like, yes, I want to create a phage therapy center. And I was like, okay, sounds like Stanford would be a great place for me to do that.
It's a great place for a lot of things.
Yes. Things happen over there. You see them all the time in the news. And I got the sense that just like, that would be good. And there will be resources and there will be support structures. And if Stanford wants to do this, I can certainly help with getting the phage side up and running and they can do the rest. And I was like, cool. So that's where I'm at, but I've just freshly started there. And so again, it's like joining a new lab. It's like, okay, what do we have? What do we not have?
You have your lab, you have your fridges, you have your vendor, you have your postdocs or your research students.
Yeah. And what I've learned here, and so this is why it's like a phase of just stepping back and being like, hey, is this going to proceed in the same way or different? Mm-hmm. And like, what is actually the need? And what do we have? And what do we do well? Because this is way more of an academic center of the standard sense. Like there are undergrad students like milling about. You can't imagine doing your phage therapy prep and checking the endotoxin level is like next to nothing. But like students are just like knocking over your media and like super excited to be part of it. And like, can I do the hands-on part? And I'm like, I need
Using your fridge, storing things in your
Yes.
fridge. One of your blog posts that I love is like, very important pro tip for your lab: buy your own fridge.
Yeah. Like, yes. So there's just, and I'm like, I want a lock on my fridge. Not because like I love the students, they're so excited. But like they just pick a vial of phage and be like, this says T4. I think I can use it because my project's about T4. And it's like, okay, I'm gonna have to start labeling things with something else to hide them. Like it's just not, there's a point where it's not compatible with academia to have an operational like mini pharmaceutical unit. And I think we got to have that at Westmead in Australia because we didn't have students. It was a research institute. We had like one PhD student, no undergrads.
Professionals there for, careers for 5-6 years. We don't have these students who are discovering how to be scientists.
Exactly. And I didn't have to mentor them, and which I really enjoyed. But also, I'm like, wait, I have a lot of time then. And like, we have the gene therapy center, and so I could like go down there, and their machine is validated, and they have a quality manager, and like.
And now you run it. But
now I have to develop the assay because we don't have a machine, and the assay needs troubleshooting, and I'm going to be the one who has to say this is safe. And like, I've had to be like, okay, this is great for a lot of things. What aspect could we get good at that fits this? And I think what it is Stanford, this lab has like a whole side on the preclinical like animal model and like delivery side. So like this is the whole area we haven't talked about, but like getting the phage to the place. That's one of the on that table of like failed trials. Right. It's kind of wrapped up in that.
Yeah, he goes to that's the like something goes to the lungs.
You can breathe it in if it's lung. You can put it on the burn wound if it's on your skin. You can do intravenous to get anywhere else. But there's like a lot of there's some data coming through that like the circulatory system will get rid of stuff first of all. And
great job with that. And plus to every pharmaceutical developer sugar.
Yes. So there's better ways, and if you can find better ways, that'll be better. And so like the joint infection, like pour it on in surgery, like maybe that's the way.
Great.
And so one of the things this lab that I'm in is doing is hydrogels, and so they have like. They work with a group that's engineering new slow release gels. So they stick to the phage and then they like release them over the course of a week. So it ends up being as if you were dosing daily, like 10 to the 9 phage per day.
But so your patient adherence is whatever. For injected use then?
This is for topical, but it could also be, they put it like they can inject it like under the skin. It could be injected otherwise, but not like in the bloodstream injected, but like locally in a sense. And maybe into the bladder, who knows, or into a catheter, who knows. But like the phages go in the gel and they come out slowly, and that like leads you to have to not treat as often for the same effect. And so that might be, we might try to position ourselves as like a center for phage delivery and get really good at like, first of all, we have the animal models all set up already, which very few phage labs have, and then like the delivery side. And maybe we do that instead of me being like, I'm making phages for each patient, and like taking one step more zoomed out and being like, I'm gonna work on this.
What's the next bottleneck? What's the like next thing that trials, fail at after not having produced the phage?
Exactly. Yeah. It's almost like, okay, I'm re-updating my operating system. It's like the next 5 years. First 5 years, we had to convince doctors. The second couple of years, we had to like make phages at all. And now I'm like, okay, someone else can make the phages at all. They know how. I blogged all my secrets are on. But like now getting them to the point where it's much easier to make them work predictably in a trial. And we're also writing grants for doing trials. And so we're writing grants left and right. That's what I do.
A licensed researcher.
Yeah. And so I'm trying to find a version of this story that resonates with whoever we tell it to, that lets us make the next little steps forward. But I'm always trying to find like, what is the actual bottleneck? And maybe it's understanding clinical trial design. Like I'm still kind of agnostic of thing. I'm just like,
why hasn't it happened yet?
Yes, why don't we have this? So I love this whole like the idea that it's not just the phage field that's like doing stuff new. I'm very interested in like soaking up all the other fields and how they're doing things differently, especially like CAR T cell, but also that's probably like the boring version of whatever's on the cutting edge by now.
Yeah. We see the stuff that is just making it.
Yeah.
Cool. Well, this has been so much fun. And I'm excited to see what it is that you're doing next, where the field of phage therapy is going next, and as we see it enter the clinic, how they or you start picking apart these problems that we think are the problems that are keeping this from helping the people it needs to help.
Yes. Thank you, Rob.
Thanks.
So fun. Aw, loved it.
We've brought together an incredible group of people who are leaders in their fields. Each are coming at this from a different perspective, but the thing that unifies them is that they believe that something can be done differently, and they're doing it.
We think we can cut this Gordian knot.
To do it smarter and better.
Our equipment is even cheaper to grow.
Then brewing beer.
Yeah.
Excellent.
Yeah.
Giving biotechs the ability to think and act more like tech companies.
These conversations are laying out the pieces of the puzzle.
The market is willing to pay for some of these premium vaccines.
Turn down the dial on the immune system that's causing it to go haywire.
We should be looking back and saying chemotherapy was barbaric.
We have an opportunity to unlock a better, healthier world.
This is not a zero-sum game.
This is Development and Research, an interview series about fixing the broken world of clinical trials.